Hello students, welcome to EPG Pathsala. I am Professor D. Velmurugan, former head of the department from Center of Advanced Study in Crystallography and Biophysics, University of Madras, Gindi campus, Chennai. I am at present working in the department as the University Grants Commission Basic Scientific Research Faculty. Today, we are going to talk on the module Crystallization of Proteins, Parameters Governing Crystallization and Data Collection from the paper Biocrystallography. Let us first consider the objectives we are going to deal with. We are just going to introduce you to the concept. Then the effect of pH and temperature on protein stability is going to be dealt with. Then we are going to look into the various crystallization methods, and also we are going to deal with crystal harvesting, mounting and data collection. The rate limiting step in the determination of the structure of a protein is the availability of good diffraction quality crystal. Many proteins do not crystallize readily, and the crystals, when available, may not be of the right quality for the X-ray diffraction work. Protein crystals are periodic self-assemblies of large and often flexible macromolecules held together by weak intermolecular interactions. Protein crystals are generally fragile and sensitive to environmental changes. In the early decades of protein crystallography, the realization that protein structures could indeed be determined successfully by X-ray crystallography was so overwhelming, and the excitement of solving the structure of the protein was so great that crystallographers resorted to trial and error procedures to crystallize protein quickly. The picture you see is taken from the cover of the journal Acta F. This shows the shapes of various crystals which are used in macromolecular crystallography. Systematic study concerning the properties of growth of protein crystals was therefore secondary to the immediate objective of determining the structure of the protein. In view of this, only a few empirical guidelines to protein crystallization could be formulated. To form protein crystals, the protein solution must become supersaturated, and in this thermodynamically metastable state, nucleation can occur and crystals may form while the solution equilibrates. Protein crystallization is a multi-parameter problem. This is the most distinction between small molecular crystallization and macromolecular crystallization. Usually in small molecules, you get crystals by using a slow evaporation when you have a compound mixed with a proper solvent. But here is a multi-parameter problem. That is why protein crystallization itself is a big task. So we will see what are all the parameters on which the protein crystallization depends in a few minutes. The search for the crystallization conditions for a protein involves performing a series of experiments in which a number of different parameters such as pH, temperature, ionic strength, protein concentration, etc. are varied. Since each protein may require the adoption of a unique strategy that may not be applicable to the next protein of interest, there is not universally applicable single strategy that would yield crystals readily in all cases. The problem may also be complicated by the presence of previously unrecognized parameters such as geometry of crystallization setup, material of the crystallizing vessels, biological origin of the protein, etc., some of which may interfere with crystal growth. Purity is one of the most important factors to look in for any crystallization effort. That's why in the 60s, earlier 60s, Nobel Prize has been awarded for mentioning or giving steps how to purify proteins and enzymes, because that is a great task. Presence of conformational heterogeneities in the purified protein, many unsuccessful or unproducible crystallization experiments are probably due to purity and homogeneity problems. It is always advisable to carry out crystallization trials on fresh samples and without mixing batches of proteins, as microheterogeneities due to post-translational modification, fragmentation, autolysis, aging effects, aggregation, denaturation, etc. may interfere during the protein crystallization process. The most common technique for protein crystal growth is by vapor diffusion, about which we are going to see soon. Many trials may be necessary to obtain well diffracting crystals. That is why determination of macromolecular crystal structure analysis is somewhat a tedious problem, and that is the reason why, even though we have in the depository more than 8 lakh small molecular crystals available, in case of proteins, the protein data bank contains about 1 lakh 25,000 entries only. Protein molecules must separate from solution and self-assemble into periodic crystal lattice in order to form crystals. One generally starts from protein solution of suitable high concentration, usually in the milligram per ml range, to which reagents that reduce protein solubility or precipitants are added. So here this also is a stringent condition, namely some protein they do not have the conventional concentration of around 10 mg per ml. So here we have to do the crystallization with robotics, where minute experiments can be easily done. Once the solubility limit of the protein is exceeded, the solution becomes supersaturated and metastable, and given favorable conditions and the presence of nucleation sites, crystals may start to grow. This figure 17.1 shows clearly the variation of protein concentration and precipitant concentration. So in the y-axis you can see protein concentration, and in the x-axis you see the precipitant concentration is just exponential decay. So now this can be divided into 3 parts. The lower part denotes the stable situation, where we have clear protein solution and in a single phase, whereas the uppermost part is unstable. This shows the 2-phase region, and we have spontaneous decomposition, precipitants plus protein solution. Clear protein solution is there. This is also a single phase. In between these two, in the middle, we have the beta-stable solutions. We have separation into saturated solution and protein. So this is an important criteria one has to understand. As it is not predictable which precipitants and conditions will yield well diffracting crystals, many initial crystallization trials have to be undertaken. Many ready-made crystallization kits are now available. For example, Hampton, molecular dimension, etc. And one can even prepare his own kit with various buffer concentration, etc. Mostly, only poor crystals are observed in initial trials, and a systematic optimization of initial crystallization conditions leading to diffraction quality of crystals follows the initial screening. The higher degree of supersaturation needed for crystal nucleation can be achieved using a variety of precipitants like ammonium sulfate, ammonium nitrate, calcium chloride, lithium sulfate, magnesium sulfate, potassium chloride, sodium acetate, sodium chloride, sodium citrate, sodium formate, sodium phosphate, which are of salt types, and there are some things like solvents like polyethylene glycol, we call it as PEG, 4000, 6000, 8000, acetone, acetonitrile, butanol, dimethyl sulfoxide, dioxane, ethanol, and methyl pentane diol, which is called MPD, methanol, and also isopropanol. So these are all the variety of precipitants used by the protein crystallographers. Among these, The most widely used precipitants are ammonium sulfate, PEG, and MPD. Sometime during crystallization, ammonium sulfate may crystallize, and people take it as protein crystals. Only after collecting data they found that it is a salt crystal. One has to be very careful. Proteins can exhibit a number of solubility minima depending on the nature and concentration of the electrolyte, concentration of the protein, pH, temperature, etc. This explains why some proteins can often grow in several different crystal forms. One example is lysozyme. Crystallization must therefore be made with a large number of sets of conditions so that one can hope to find one particular solubility minimum that yields crystals. Protein sample available is usually very small, and hence one should first get some feel for the precipitation behavior of the protein. This is called some trial experiments. A series of simple preliminary experiments can be performed on a macro scale, and observing precipitation under microscope can conserve the precious material and narrow down the initial range of likely conditions. So now we are going to consider the effect of pH and temperature on protein solubility. Since the protein is a labile molecule, all the extreme conditions of precipitation, pH and temperature should be avoided. It is much easier if crystals are grown in conditions not too far from the physiological environment in which the molecule operates. The pH value has a strong influence on the solubility, which has a minimum at a pH close to the isoelectric point of the macromolecule, which you are going to see from figure 7.2. The ionic strength can have the opposite effects on the protein solubility. That is, solubility decreases exponentially with increasing ionic strength, a phenomenon known as salting out. But it also has a minimum at a very low ionic strength, the salting in effect. Precipitation can be achieved by increasing the salt concentration or by dialyzing the solution of protein against water. The pH of a protein exerts very strong effect on protein crystallization as evidenced in the figure 17.2. So this is a figure we have been just mentioning about the variation of the protein solubility, which is shown in the y-axis as a function of the pH of solution, which is shown as the x-axis. If you look here, the exponential decay, the protein solubility comes down, reaches the minimum at a point known as isoelectric point, then it increases. Of course, isoelectric point is the one at which the net charge of the protein is 0. So usually at this point, isoelectric point, the pH is around 6.5. Although the solubility minima correspond well with the isoelectric point called Pi, the pH at which the net charge of protein is 0, the correlation of Pi and the actual pH of crystallization is weak, which means that proteins do not crystallize best or most frequently at their Pi. The strong effect of pH on the crystallization success is likely more a result of affecting the total charge distribution than a net effect of the pH on protein solubility. The protein solubility can either increase or decrease with temperature. Most proteins are crystallized either at room temperature or at 4 degrees centigrade. The reason for the choice of this specific temperature arises from the attempt to slow down the degradation by proteases. Individual proteins may crystallize better at a certain temperature, and a lower temperature can be helpful to slow crystal growth or to change the dominance of certain crystal form. The reasonably constant temperature level should be of advantage in principle simply to keep stable whatever crystals may have formed. Now we are going to see in detail the vapor diffusion techniques, where we are going to deal with hanging drop and sitting drop, the conventional methods for protein crystallization. Vapor diffusion is the most popular manual technique for growing crystals. I am specific about manual technique because now robotic crystallization has become possible. Here, the unnecessary wastage of the protein solution or buffer solution is avoided, and minute sample can be handled in a more efficient way. A few microliter of the protein solution is mixed with an equal amount of reservoir solution containing the precipitation reagent cocktail. In hanging drop vapor diffusion method, a drop of this mixture is placed on siliconized glass slide which covers and seals the reservoir. The advantage of siliconized glass slide is that we can invert the glass slide and drop will not fall. The reservoir solution is usually more concentrated than the protein precipitant mixture in the hanging drop, and hence water evaporates from the drop into the reservoir. So this setup is shown in figure 17.3 next. As a result, the concentrations of both protein and precipitant in the drop slowly increase until the protein solubility limit is exceeded. At this stage, the solution becomes supersaturated and nucleation and phase separation occur, leading to protein crystallization. Figure 7.3 shows the vapor diffusion method technique. As you saw already, the droplet shown in the top with an arrow mark contains the cocktail and protein solution from which protein crystals can be obtained after supersaturation. The bottom arrow indicates the crystallization cocktail containing the precipitant, additives, detergents, salt, etc. So now, the next slide, 17.4, shows a set of wells called Limbro plates, where in each well you can use different protein crystallization setup by varying the parameters, and you can mark them on the top of the cover. So what we have seen on the right hand side of the earlier figure in the Limbro plate contains 24 wells, and these are mostly used for hanging drop vapor diffusion technique. So these plates are polystyrene plates, and these are relatively cheap, and each well can be individually sealed with a siliconized glass slide, and many crystallization trials can be attempted. The drops are easily observed under the microscope. A variety of other crystallization setup kits are also available. So now you are going to see an interesting video showing the robot crystallization. You see here a conveyor belt like thing is running from which is micropipettes. This is like a plastic, which has been used and thrown away. It has been cut and used to fill up a column in the plate. Simultaneously, we can do many crystallization trials, and this micropipetting is done to take the buffer solution and protein solution, everything, and crystallization drops are being set up by the machine. So it can play with very low, suppose you have the protein of much lower concentration, lesser than 10 mg per ml, these cases robotics can be used. We have a system called Mosquito, which comes about a crore, which could be used for the automatic protein crystallization. In sophisticated labs, mostly people use this robot crystallization for crystallizing the protein. It is very efficient. You can just see how the robot picks up, cuts the micropipette, then takes the buffer solution, everything, how it sets up the drop, and it also do the covering of the plate, which can be set for crystallization. So here we are going to talk about the sitting drop. The principle is same, only difference is instead of the drop hanging, now it is sitting. The basic methodology of sitting drop method is the same as that of the hanging drop method, which is a slow evaporation technique, except that the drop is now replaced on a shelf and it is comfortably sitting. So this is shown in the figure 17.5. The lower arrow mark shows, again as usual in the hanging drop, the crystallization cocktail containing the precipitants, additives, detergents, salts, etc. And in the sitting drop, which is made up of the cocktail and proteins, crystals will be formed. The sitting drop method is automated and many 96 well-played variants are now available. The limitation in the sitting drop is that there is a difficulty in harvesting the crystal from the bottom of the drop than from the removable cover slip, as found in the hanging drop method. All the drops are set up at once using robotics and then the plate is sealed with an adhesive. Let us now consider the match method. So what is the batch method? There are some general methods of crystallization of proteins and batch method is one among them. The other methods are bulk dialysis and micro techniques in addition to the generally followed vapor diffusion technique. Batch method requires a large amount of protein sample. In this method, an appropriate precipitant is added to the buffered protein solution until the solution develops barely visible opalescence. The solution is then centrifuged and are filtered and set aside for few days or weeks. This method often requires high protein concentrations of 10 to 100 mg per ml and involves screening with various buffers, salts, precipitants, pH, etc. in vials containing 0.5 to 1 ml of protein solution. This method often yields microcrystals that can be used for seeding. The first efforts in the use of micro techniques for proteins available in small quantities were made by Zippensauer in 1953 using the procedure currently called as microdialysis. Protein solutions of less than 100 microliter are taken up in cells made of capillary tubes closed at their ends by dialysis membrane or polyacrylamide gel plugs. The entire assembly is then submerged in a vessel containing the solution of concentrated salt or organic solvent. Slow equilibration takes place across the membrane and crystallization occurs frequently. So now we are going to look into the seeding technique. As the name indicates, seeding, we are putting as input some crystals. So seeding is generally resorted to when efforts at subsequent crystallization could not yield any further crystals for whatever reason, because of multi-parameter problem. We cannot find exact reason for not getting protein crystals. Seeding also helps to minimize the quantity of protein needed during crystallization trials. Here, one transfers microscopic crystals to a non-nucleated protein solution. Occasionally, one can also crush bigger crystals to micro pieces and use them as seeds. These procedures are called micro-seeding. Let us now consider the membrane protein crystallization. If one looks into the protein data bank where the entries are deposited for whatever protein that have been solved, the number of proteins related to membrane proteins is very much limited. This itself tells you that solving membrane protein search is a difficult problem because the crystallization of membrane protein itself is a tedious problem. That's what we're going to see now. In membrane proteins, the hydrophobic surface is exposed to lipid bilayer and the hydrophilic region towards the solvent. In order to solubilize them, detergents have to be used. During the extraction and purification of membrane proteins, some phospholipid molecules are bound to them, leading to successful crystallization. In type 1 crystals of membrane proteins, the membranes are organized in membrane-like layers stacked one on top of the other. These crystals can be grown starting from a lipidic cubic phase or from basiles. Sometimes they also grow from normal detergent solution. Growing from basiles is much simpler. In type 2 crystals of membrane proteins, macromolecules in the lattice are in contact through hydrophilic surfaces and they usually contain large amount of solvent. Use of antibodies against the protein of interest in membrane protein crystallization is totally a different approach. This method works in the case of proteins with a large hydrophobic surface which can be masked by Fab or F5 antibody fragment. The real drawback of this method is a very time-consuming procedure for preparing monoclonal antibodies. So we are going to now give few guidelines for protein crystallization to help achieve the goal of crystallization, and these are listed below. 1. The protein must be pure. 2. The solvent chosen should not denature the protein. 3. The higher the protein concentration, the better is the crystallization. 4. One should be aware of the batch variation in the protein sample. 5. Protein solution must be centrifuged and filtered. 6. You have to work in a clean lab free of dust. And last point, namely 7. Precipitating agent should be added extremely slowly to avoid amorphous precipitation of the protein. In recent times, most of the labs use crystallization kit and robotic crystallization for the above purpose. After these 7 points, the most important one is point 1, the protein must be pure, because only pure proteins will give good diffracting crystals. Only good diffracting crystals will yield good data, and we are starting the protein crystal structure analysis only from the X-ray diffraction data. Ultimately, the model you propose from these data depend on how pure is the protein. So that is why, even if waste a lot of time, it's good to prepare the protein in the purest way. Now let us deal with the harvesting of crystal, the mounting of the crystal, and data collection using the protein crystals. The protein crystals are delicate and hence proper harvesting, cryoprotection and mounting have to be done for successful data collection. For a good quality data, the first step for successful data collection is the careful mounting of the selected crystal without any damage. To avoid the drying out, the crystals need to remain in the mother liquor during the data collection process. In capillary mounting, the crystal, viewed under a microscope, is sucked up together with mother liquor from its growth drop into a thin-walled quartz glass capillary of suitable diameter. The capillary is sealed after wicking out the liquid with wax or some inert glue and centered on goniostat for data collection. In cryomounting, a single crystal is scooped out of the mother liquor with a nylon loop or microfabricator capped and mount attached to a steel pin with a magnetic base. This captured crystal may directly be dipped into liquid nitrogen if there is a sufficient cryoprotectant already in the crystallization cocktail. The crystal is briefly swished through a cryoprotection before quick dipping into liquid nitrogen. Common cryoprotectants are glycerol, ethylene glycerol, MPT, sucrose, and low molecular weight PEX, all in a buffer compatible with and at the same pH as the crystallization cocktail. Special storage cylinders to export the crystals to synchrotron data collection centers are also available to conserve the crystals permanently. Now, from our country, we have some lease lines in these synchrotron beams in Europe. Data collection at nearly 100 Kelvin prolongs the crystal lifetime by a factor of nearly 70. There is an advantage in radiation damage, and this is known as rip phasing, and also the crystals diffract to increase resolution under cryogenic temperature. For data collection, particularly on synchrotron beamlines, user support is generally quite efficient. Various standard strategies are implemented, assuring that a complete data set will be obtained. The crystal is generally rotated in small increments of 0.1 degree to above 1.5 degree when exposed to X-rays, and reflections are recorded on an area detector. The low resolution part of the diffracting data is important for phasing, and the high resolution data help model building and refinement. With the present day advancement in molecular biology experiments and also in the electronics, nowadays many structures are solved at atomic resolution or near atomic resolution. Thus, there are no unimportant parts of the data that can be neglected during the data collection. So, students, let us summarize what we have learnt in this module. We learned that protein crystals are periodic self-assemblies of large and often flexible macromolecules held together by weak intermolecular interactions. For a protein to be crystallized, the protein must be supersaturated in a suitable buffer, and it follows a saturation curve to form crystal passing nucleation phase. The crystallization of proteins is a multi-parameter problem in nature. Purity and homogeneous nature of the fold of proteins are very crucial parameters in protein crystallization. The most commonly used technique in crystal growth is vapor diffusion method. A broad variety of precipitants are used to achieve crystal nucleation, among which polyethylene glycol, PEG, and MPD, ammonium sulfate, are mostly employed. Apart from protein purity, pH, temperature, and concentration of the protein and the precipitant, they play an important role in the crystallization of proteins. Seeding technique is employed for crystals to grow bigger and better in quality for better diffraction. Suitable cryoprotectant is used as the protein crystals are fragile during data collection. Thank you.